Okay, welcome everyone to the last of our 2020 IMAG MSM working group meetings. I hope the individual subgroups will continue to meet, but this is our last plenary meeting, mini seminars and discussion. Today we have two mini seminars by Reed and Liliana, and then by Filippo. These are, of course, our short 15-minute teaser presentations, hoping to excite new collaborations and new ideas. I need to remind you that the meeting is being recorded. It's being streamed, live streamed, and it will be made available as a recorded document. So please bear that in mind. Myself, James Glazier, co-lead with Reinhard Labenbacher, web administration by Jim Sluka. You've all had coordination information by Bruce Shapiro. We should probably add Juliano to the list because he's been handling Twitter and Slack issues. Remind you, we do have a Slack channel if people want to use it. It's available. We can also post information, and we encourage you to post information on the IMAG MSM wiki pages. So please contact us if we need any help with either of those communication methodologies. Then without more ado, we will start with a brief presentation and discussion by Reed and Liliana from NIAID, and I will turn it over to them.
Great, thank you. So Liliana is on the call as well. My name is Reed Shabman. Can you all see my screen? Okay, great. So I'm going to try to stick to time, and I'll be giving the overview. Liliana and I put together a few slides, and she may jump in. But I wanted to say thanks again, and just I'm appreciative of the invitation to present. And today I want to talk about data dissemination and modeling infectious diseases from a NIAID perspective. So both Lilian, Anna, and I are in the Office of Genomics and Advanced Technologies. So the quick presentation overview, just my personal opinion and our office opinion on goals and challenges of multiscale modeling. You all are the experts, and so this is from a program officer standpoint. I then want to highlight one program, the Systems Biology program, where this is a consortium that does some modeling activities, and highlight these just to make folks aware of individuals in the field that are doing some of this work. Give a quick perspective on data generation and deposition, and then jump into some discussion if anyone's interested. And unfortunately, I don't have a ton of time to go too deep into each of these, but just try to touch the surface. So we kind of see the goal. So when we think about multiscale modeling of infectious diseases, I've tried to highlight here goals and challenges. And as you can see, the keywords are kind of the same. So in our mind, goals are to integrate clinical metadata, large data sets, and cutting-edge technology in the context of infectious diseases, and apply these findings to predict disease outcome and inform treatment options. Ideally, we'd be able to validate and obtain mechanistic insight from this data integration, validation through laboratory studies, perhaps from clinical specimens where you have most biologically relevant yet imperfect data sets, and then have the ability to refine these. And I think on the flip side of the coin, the challenges are really, there are challenges with data integration. How do you integrate different types of data? How do you validate and reproduce model results both within your own group, but maybe across groups and geographically spaced? And then finally, how do you refine models to actually improve accuracy? So can we predict survival, or can we predict binding interactions with greater accuracy or greater forecast? So this is a slide that I borrowed from Michael Yeaman at UCLA, who's part of the Systems Biology Centers that I will highlight it here in a second. And this is really just to highlight the fact that we think of these different scales of disease of modeling. So you can model all the way from in silico to in vitro, all the way up to clinical models. And really, each one of these has a place. Each one of these is important, and each one of them has their own challenges and ways to go. So it's something that we think about often. And just drill down into specifically where are we sit in this, or who are some of the individuals that our office supports in this effort, is the Systems Biology Consortium for Infectious Diseases. And so this is what I'll be talking about for the meat of the talk. This is a community that integrates experimental biology, computational tools, and modeling across time and space to develop strategies to predict and alleviate disease severity across multiple pathogens. So there's 11 centers in total. They will focus on antimicrobial resistance, tuberculosis, influenza, as well as emerging infectious diseases. And the point of this talk is to highlight that they put out individual papers around these infectious diseases, but that we also have a working group, the modeling working group, within the centers that try to share ideas and efforts in those areas. My slides are not advancing. Okay, there we go. So the overall accomplishments of this modeling working group, and I can provide these links later if you're interested, but there's a total, it was formed in 2016. There's a total of 14 presentations on different modeling topics, each with slides and tutorials and code available. So these aren't really like seminars like I'm giving now. They're more the nuts and bolts of Jupyter Notebooks and how you actually might do a beast analysis. Workshops have been done before by John Monk and Bernard Paulson, where they looked at genome scale modeling at our annual meeting, and the slides are available online. And a number of collaborations have come out that have fostered publications. So this is a productive group. So, you know, UCSD and Baylor have joined teams, Sloan Kettering and UCSD, for example. So it's brought together individuals from across the different modeling spaces. And really, I mean, I think this group doesn't need to see this slide, but we do want to address complex questions through data integration and modeling. So we're interested in all different types of data: clinical data, genomics, transcriptomics, microbiome, proteomics, metabolomics, lipidomics, like glycomics. How do we integrate all this data to make outputs where we can look at pathogen host networks, predict patient severity, develop software, harness patients' gut microbiome to cure infection and understand that a little bit better, and then to maybe advance drug treatments. And we're also very bullish about putting data into repositories. So I've highlighted just a small number here. These are NCBI, dbGaP, GEO, for example, and then also bioinformatics resource centers, which are also managed in the office where Lilian is the director, which is the BBVRC and VUPath DB. So I'll just give a couple examples of different models that have been, different outputs that have come from this program. And if you're interested in looking at these papers in more detail and reaching out to the authors, that'd be fantastic. So maybe some in vitro and silico approaches here are some examples of how you would model complex data sets. The Boston College group has looked, Tim Van Option has looked at evolutionary pathways to AMR in test tubes and both in planktonic and biofilm conditions. So here, not viruses, but are able to predict antibiotic evolution in vitro. Bernard Paulson's group has done a lot of work, and I just highlighted one picture here, which I can't fully describe, but they are really looking at how do you layer different types of data, right? So how do you take GWAS data and metabolomic data and layer it on to start looking at AMR phenotypes, where you can look at resistant strains in red versus susceptible strains in blue when you're looking at both metabolic states and genome-wide associations of SNPs, for example. So it's ongoing work as well. And you can, on another scale, you could look at models of disease severity and survival. There's a number of examples. Here's two. One is from the Sloan Kettering group. Joao has shown that microbiota can be a predictor of mortality in allogeneic hematopoietic stem cell transplantation. So they essentially are showing that higher diversity of microbiota post stem cell transplantation in cancer patients results in a longer survival or a higher level of survival in patients compared to a lower diversity of the microbiome. And they're able to develop models that are going to predict survival in these categories. You can also look at this in the context of Staph aureus, so mortality risk, profiling of Staph aureus bacteremia by multi-omic serum analysis. So they're looking at serum and taking a number of large-scale data sets to predict early and predictive and pathogenic signatures. And then finally, I think important for this group are pandemic response efforts. So can you model PPF, protein-protein interactions. Nevin Croman's group has looked at this with built networks of SARS proteins and then done molecular docking of antivirals, for example. And they visualize these in index, so you can look at these protein networks. And also, Doug Laufenberger's group has performed system serology, and Geladoctor's group has done system serology to look at distinct serology signatures that are associated with prediction of individuals that will recover versus individuals that might succumb to COVID. And so that was a kind of a whirlwind, but just to point out that these are the types of data that are being generated and the types of outputs that are being generated from the systems biology centers. And I think one critical question that we think about every day, if not every hour, is how do you get data out into the public space so that it can be used for further analyses, either primary or secondary analyses. And so we made a flyer, or Grace Teets, who I think is on the call here, and Liliana made a flyer a while back, and it's called 3 Easy Steps to Share COVID-19 Data. And I put ... or any other infectious disease data sets. And it's very simple. It's to identify a specialized or general repository, deposit your data set, and this can include process data, and then make sure you add the appropriate metadata, because that's very powerful. There are a variety of different repositories that could be used. And again, COVID-19 data, this could be interchanged with any data, right? But there are specific COVID-19 hubs or SARS-2 hubs within a lot of repositories if you're interested in specifically that data. And so this would include things for reagents, BEI, structure, omics, immunology, assays. And if you go to the website, you can get a whole list of some of these repositories. And then you can also deposit into generalist repositories. So on the previous slide, these were specific repositories. Generalist repositories such as Dataverse, NIH Figshare, these are all places that you can deposit data in any format that you like. And I think that doesn't solve the problem, but I think it's important to get data out there. And then there's a challenge of identifying metadata and data to leverage for multiscale modeling. The Systems Biology Consortium is also trying to aggregate data. So Outbreak.info is a great aggregator of disparate data into a single website. And so you can start to look at different types of SARS-CoV data, and maybe you can find information there that might help with some of the analyses that you're doing. And this is an ongoing effort, as well as generating sort of a searchable digital collection of systems biology assets so that others in the community could identify those. And so finally, I highlighted one program. There are a number of programs, other programs that are data generators that one could leverage for multiscale modeling. This involve CREED, SEERS, and CIVICS. These are viral programs. SEERS and CIVICS have an influenza focus. CREED is emerging infectious diseases. There's an RFA Adam pointed to here that's undergone review, and this is for the antibiotic-resistant bacteria. Clinical units such as the CIVICS again, and the VTEU generates data, and these centers can develop and improve animal models, develop reagents. They're establishing longitudinal cohorts, and they're expanding human challenge study capability and capacity. And so I would say thank you and take any comments or questions. That was kind of a high-level overview of our perspective of things. But if anybody wants to talk more about data, deposition modeling, specific modeling efforts or future collaborations, you have to.
Okay, thank you very much for the overview. That was impressive, comprehensive in a short amount of time. The slides and information will be available for people to refer back to. So we have time for a few questions.
This is John Rice. And how much involvement do you have from the Zoonotic Science side? You have the veterinarians and the animal scientists who are actively getting interested in what's coming out of the human research that they could use?
It's a great question. Lilian, do you want to have any thoughts?
Well, I would say, for example, when we had cases where there's zoonotic transmission, we have had people in that space. So the group in Tulane that collaborate with the Systems Biology Center for Emerging Viruses at Scripps, they have a group of veterinarians and other people who would be more studying environmental transmission, or not environmental, but zoonotic transmission into humans. And then through our Sears networks that Reed talked about for influenza, we have a large surveillance effort in waterfowl and other types of environments where you could have the admixture of the viruses that would then jump over to humans. So I think there is involvement from the types of people who would be generating this type of data.
But it's not as strong. It's an area that we might explore improving. True?
Yeah, I think in general, not only zoonotic transmissions, environmental exposures as well, and how do we detect things in the environment, and whether there's any thresholds that would initiate some sort of alert or something for infectious disease surveillance. Yeah.
Thank you.
Reed, this is Reinhard. Can you maybe say just a few words about the grant program that supports these centers?
Sure. These are, let me just step back. Yeah, so just to add on to Liliana's comment, Bob Gary is one of the, is in that systems biology grant that Liliana was referring to, and they're looking at rodents and the reservoir hosts for Lhasa, and so in West Africa. And so that's an interesting, but I think there is more to be done there. These are, the systems biology program was established in, around 2008. I'm gonna let Juliano correct me in a second. But there's been a number of iterations. These are they started as contracts, they're now grants. They're UO1, U19 programs in 5-year intervals. And currently the latest iteration of that U19 is around, it's about halfway through. And so, the office that I'm in now has recognized these holistic approaches with large data and modeling as an important attribute and a gap that needs to be filled from a consortium level, because we think that folks are from different backgrounds mixing back are better together than apart. And so that's kind of a high level.
Good, thank you.
I'm looking to see if there are any other questions. I'm always intrigued personally because one of the things I have as a sort of a slogan is that ultimately properly annotated data should be able to be turned into executable models, and models themselves, if they're annotated properly, should be able to embody knowledge and exist as data. We're a long way from being able to do that, but I have a personal belief that we need to try to bridge the gap which we see between models and data, and to try to merge those into something that's more unified. It's not an easy problem to deal with, but you maybe have thought about that.
We have, and I think our groups have talked about it quite a bit, both internally and extramurally. So I'd say one of the concepts and ideas that we're trying to really push is to get people to deposit not only the raw data from their experiments, but also the process data after they have done their cleanups and everything else, right? Because so much of the downstream effort really depends on how you do that first step. And many times that process data, then analyzed data is only available in a table in a paper. And so you don't really have that in a computable format. So we're trying to really address that and making sure, and we post there in our handout said, not only just your raw data, but also your process data. So that's something that we're trying to convey to the data generators, that is imperative and important, and also to make their workflows and their protocols of how they actually process the data also available and ideally reproducible, right? So we had a really interesting conversation with the editors of eLife, who are really trying to make sure their articles are fully reproducible, right? And so that was, we were already talking about that, and then we had them give us a talk at the, I think it was, I don't remember which one of our working groups, but really that's the objective, because I think data on its own is good. Data with all of the other elements and process data is even better. And similarly with the models, we've had a lot of conversations about what's the best place to deposit the models, how do you deposit, how how much annotation and do you put into the models themselves? And so these are all ongoing conversations that I'm sure you're having here, and we're having similar conversations in our working groups.
I'd love to talk more about that at some point if you like. So, and I know there are many people in this community that are interested in aspects of this. So that's great. Is there time for one last question before we move on? There's so many things that were raised as interesting.
Yeah, if I can just ask a question. So my name's Mitchell. I'm a graduate student at North Carolina State University. I just want to ask, from your perspective, do you approach the machine learning kind of strictly data-driven approach in a different manner than mechanistic modeling when you're looking at funding and grants and what you're trying to get done with your organization?
So do you want me to take that, Liliana, or should
Go ahead, Reed.
all right, I'll say, so the short answer is we're interested in all different types of models, whether that's pure computational or if there's a lab component or a mechanistic component. The bottom line is there's also an Office of Data Science that is pushing a lot of things, and there's actually a council approved concept out there that's specifically highlighting just novel approaches for computational modeling. So we can drop that into the link. It's not a foe on the street, but it's a publicly available concept that's been approved by NIAID Council. Now, but going back to your original question real quickly, Bernard Paulson is kind of a leader in our systems biology with a lot of this type of computational modeling from just data, secondary data analysis, and we think is an important component.
Great, thank you.
And Reed, a quick request. Our working group has a subgroup for dissemination, education, training specifically as a subgroup. And follow up to this, I'd be really interested in hearing some suggested names of people who know what you're doing, who might have a little bit of time to be part of another working group and help us not be reinventing wheels. And so if you have a candidate in mind, please share. Thank you.
Okay, thank you very much. I'm sorry we have to move on. Thank you again. I hope we can have you back and talk more in the future. Really appreciate your participation. We're going to move on now to Filippo Castiglione, who's staying up very late to speak to us from Rome.